Hello students, welcome to the EPG Pathshala. I am Dr. Imtiaz Hassan from Jamia Millia Islamia, New Delhi. Today I am going to talk about the protein sequencing and concept of protein evolution from the paper Biomolecules and their interactions. So the objective of this module is to discuss the following things, like the first we discuss about the what are the mechanism or what are the methods used for the N-terminal sequencing and the C-terminal sequencing. There are two ways to sequence the protein. One is the starting from the N-terminal side, means amino side, and the one is the C-terminal side, means the carboxyl side. But most widely used technique is the N-terminal side, although there are some techniques available for the C-terminal sequence determination. Then we'll discuss about the protein fragmentation, in which especially for the bigger protein, if you want to sequence the bigger protein, it is very difficult to analyze the data. So first, this bigger protein is broken into the different fragments by using the different enzymes, especially trypsin and chymotrypsin, which cleaves at the specific residue. So we will give you the example that what are the different enzymes used for the fragmentation of polypeptide chain. Then for the intermediate sequencing, we discuss in detail about the Edman degradation method and the importance of overlapping sequences. Like if you have the long polypeptide chain, so it first cleaved into the different fragments, then sequence of each fragment is determined, then how these fragments are overlaid over each other to analyze the exact sequence of the full polypeptide chain. Then we'll discuss about the mass spectrometry technique that is used for the sequence determination of a polypeptide chain, and then at the last we will discuss about the how protein sequences help you to understand the evolution process and the function of the protein. So all things we will discuss in detail here. So let's start with the first method, is the method for the protein sequence determination, and that is the classical method, that is the Edman degradation method. In the Edman degradation method, as we discussed, that we take the Edman degradation reagent fluorodinitrobenzene, and that it binds with the N-terminal site or amino groups, and then it weakens the first peptide bond, and ultimately it is cleaved easily by the acid hydrolysis. So this reaction or this cycle is repeated many times, and ultimately each amino acid released one by one, and then this amino acid are identified by HPLC. So ultimately you can identify each residue one by one, and it will give you idea that what are the sequence of each residue. Ultimately you will be able to identify the sequence of a polypeptide chain. Then we will discuss, then the other way is to the mass spectrometry. It is the modern method and it is a widely used method. There are two types of mass, like the MALDI MS, means the mass matrix assisted laser desorption ionization mass spectrometry, and another is the ESI, that is the the ESI mass that can precisely identify the mass of a polypeptide and thus the amino acid sequence. And the advantage of mass spectrometry is that you can identify the bigger proteins as well, and it is based on it is provide you the data with the high accuracy, and then it is also utilized for the identification of post-translation modifications such as the glycosylation, meristylation, phosphorylation, etc. And the best way is to determine the amino acid sequence is from the DNA sequence, like because the DNA sequencing is a little bit easier method as compared to the protein sequencing. So DNA sequence can be easily analyzed, and then the DNA sequence using the genetic code is translated into the protein sequences, and that protein sequence is used. Here we discuss about the first sequence or the first protein which was taken for the sequencing. So the first protein was the insulin, because it is the smallest protein, and you can see here the structure of insulin. It contains two chains. One is named as alpha chain, that is the longer, and one is the alpha chain and one is the beta chain. So beta chain is longer and alpha chain is shorter, and there are three disulfide bonds are present. One is the intramolecular disulfide bond within the A subunit. However, the two intermolecular disulfide bonds are present that connects A and B chain together. So this is the simplest protein that was taken for the sequencing studies. We show you that how the sequencing was done. You can see here that first the disulfide bond should be broken, and this is broken by the two-way, either by the reduction with the dithiothiotol or by the oxidation of using performic acid. You can see here, while oxidation by performic acid is a very strong acid which cleaves the two disulfide bond and it forms the sulfonate species. On the other hand, the DTT, which is a compound that also having the free SH group, once it reacts with the disulfide bonds, it forms disulfide bond within them and cleaves the disulfide bonds of the protein and release both the species separately. Once you want to know how many number of amino acids or what is the actual composition of amino acid in a protein, so it has to be determined by the acid hydrolysis. Earlier I shown you that first the disulfide bond should be cleaved and then it is utilized for the acid hydrolysis studies. Once it is necessary that the break disulfide bond, otherwise the two cysteine residue still get attached. So to get the amino acid composition, first the whole protein should be broken, disulfide bond of the protein broken, followed by the acid hydrolysis. There are many ways to do the acid hydrolysis, but the most frequent use is the 6-normal HCl, and it is used for several hour heating at the 100 to 105 degrees Celsius, which hydrolyze peptide bonds completely. You can decrease the temperature and increase the time, or you can increase the temperature, decrease the time. Although it is a most useful method, but there are so many disadvantages of this hydrolysis. First, that the acid hydrolysis by using 6-normal HCl destroys tryptophan residue. It is a bulkier residue and it is very easy to destroyed by the 6-normal HCl. Another problem is that it partially or slowly destroys hydroxyl containing amino acids such as serine and threonine. There are another problem with this. Because of the highly acidic condition, it converts asparagine into the aspartic acid or similarly glutamine into the glutamic acid. So in the analysis, the glutamine and asparagine always comes with the asparagine and aspartic acid and glutamic acid. So that sometimes creates a problem for the identification of the composition analysis. And another fourth problem is that it slowly hydrolyzes peptide bond between the vicinal, isoleucine, lysine and valine. So although this is the best technique, but they have some disadvantage that you must know. It deals by the KOH hydrolysis to determine the tryptophan content. Then acid hydrolysis over 2 hour, 4 hour, 6 hour for the serine, threonine, isoleucine, valine and leucine. And it measures ammonium ion to be determined the amount of N plus Q given by the D plus E. It means that we should consider that the amount of ammonium ion released that is corresponding to the amount of asparagine converted into the aspartic acid and glutamine converted into the glutamic acid. So the sum of the asparagine plus glutamine equals to is taken for the amount of ammonia released plus the aspartic acid and the glutamic acid. So these are the some ways to overcome with the acid hydrolysis problem. How the amino acid quantified? So far we discussed that first you take a protein and the sample protein is the insulin, then its disulfide bond was broken, followed by the acid hydrolysis. Once you hydrolyze, then you have to quantify the each amino acid separately. Since amino acid is the colorless, so you can't analyze it in the easy way. So the best way is to take the ninhydrin solution, and here you can see the chemical reaction taken. This is the structure of ninhydrin. Once it comes in contact with the amino acid, the NH3 groups forms a reaction with the ninhydrin and it releases the purple color compound. So this purple color compound is corresponding to the different amino acids. So this compound is actually having the amino acid content of the different amino acid. So the intensity of the production of the color compound is directly proportional to the amount of amino acid. If so, for that you have to run the standard of the ninhydrin derived amino acid, and then you can quantify that the different amino acid composition in the particular protein. So it is important thing here is that the ninhydrin is colorless, but it produces an intensely purple color when it reacts with the amino acids. Once you got the ninhydrin derived amino acid, so how to quantify the amino acid? This peak is showing, this is the actually the elution profile of the standard amino acid using any technique. Basically people use the ion exchange chromatography, or even you can HPLC. So you can see here the, for example, a peptide of the sequence containing alanine, glycine, aspartic acid, phenylalanine, arginine, and glycine. So you can see here the position of the different amino acid is given as a form of a standard, and you can see here the peak of aspartic acid, peak of glycine, peak of alanine, peak of phenylalanine, and peak of arginine. So you can see here, since all these amino acids are present singly, so you see the peak intensity or peak height is the having a particular level. However, you have two glycine here, so the glycine peak is, you can see here, it is the doubles in the intensity as compared to the other amino acid. So it indicates that the position of peak is corresponding to the types of amino acid, and the intensity of peak corresponds to the number of amino acid. So if you have the standard of number of amino acid, you can easily quantify that what are the proportion of the different amino acid in a given peptide after the hydrolysis. We show you that how the amino acid sequencing was performed. So far we discussed that how to determine the amino acid composition in a peptide, and you can see that it can easily be determined by the acid hydrolysis followed by the quantification with the using ninhydrin. But if you want to determine the amino acid sequence, it is a very difficult task, and although there are several techniques are available. So first we classified into two groups, like the N-terminal sequencing and C-terminal sequencing. So here we first discussed the C-terminal sequencing, and the C-terminal sequencing was done by using there are two methods for the C-terminal sequencing. First is the enzyme based C-terminal sequencing by using the carboxypeptidase enzyme. The carboxypeptidase enzyme specifically cleaves the C-terminal residue. For example, you can see here that the carboxypeptidase A, which was derived from the bovine pancreas, and it cleaves, however it have a specificity to the arginine, lysine, proline. Then the carboxypeptidase B, it is derived from the bovine pancreas. Carboxypeptidase C, it is derived from the citrus leaves, and it used for the all free C-terminal residue at the optimum pH of 3.5. So then carboxypeptidase Y, it is derived from the yeast, and it is used to cleave the all C-terminal residue but slowly with the glycine. Then leucine aminopeptidase derived from the porcine kidney, and here it is unable to cleave the proline. Then the aminopeptidase M, it is also derived from the porcine kidney, and it is used for all free N-terminal amino acid residue. So carboxypeptidase is actually carboxypeptidase A and carboxypeptidase B is unable to give you to cleave the arginine, lysine, and the proline residue. I will show you the another technique used for the C-terminal peptide analysis, and the C-terminal peptide analysis using the hydrogenolysis. So for that, you can see here it is a sample peptide, and then once it reacts with the hydrazine solution having the two nitrogen groups attached by covalent bond. So hydrazine solution is very easily to react with the carboxy terminal of the amino acid, and then it forms the hydrazine derived amino peptide, and then after the amino acyl hydroazide are used, so it cleaves the C terminal amino acid easily, and you can see here the free amino acid are liberated. So now this liberated amino acid can easily be quantified either by the HPLC or by the paper chromatography, and can easily be identified that what are the actual residue at the C terminal site. So polypeptide plus anhydrazine at 90 degrees Celsius, followed by mild acid ion exchange resin for 20 to 100 hours, and you can get the free amino acid. So you can easily determine what are the residue present at the C terminal side of a peptide. I will show you that how the N-terminal sequencing was performed. So it's a very easy thing and widely used technique still in all the labs. So there are two reagents used for the N-terminal sequencing. One is the fluorodinitrobenzene. Here the two nitro group attached with the benzene, and the another is the dipsyl chloride. So you can see here the dipsyl chloride can easily react with because of the presence of SOCl group, it can easily react with the N-terminal amine group of the polypeptide. Once it attaches to the N-terminals of the polypeptide, it weakens the first peptide bond as compared to the other peptide bond. So once you do the mild acid hydrolysis, only the terminal peptide bond is broken and not the other peptide bond. So it can easily leave the N-terminal peptide and release the dipsyl. For example, here the first residue is the alanine, so it released the dipsyl alanine. Similarly, the dipsylated amino acid is released for the different amino acid, and this dipsylated amino acid is a fluorescent pigment and fluorescent in color, so can easily determine by the elution using through the HPLC, and then the people have the standard of the dipsyl chloride derivatives of the amino acid, so it can easily be identified that what are the residue located at the N terminal. This reaction is again followed, so you can see here the second residue is the glycine, third residue is the aspartic acid, fourth residue is phenylalanine, fifth is the arginine. So in the same order, the arginine first, the alanine will release, then glycine, followed by arginine. So one by one this cycle proceeds continuously, and each amino acid released one by one. In the same way, you can determine that what are the actual sequence of the polypeptide from the N-terminal site. I will show you the another method for the N-terminal sequencing, that is the Edman degradation method, and this is the most widely used method. Here the phenyl isothiocyanate are used. So phenyl isothiocyanate are reagent which can be used for the determination of N-terminal sequencing. You can see here that the phenyl isothiocyanate reacts strongly with the amine group of the polypeptide. You can see here, because this carbon is attached to nitrogen and sulfur, so it is the electron deficient species. So at high pH, the NH3 groups attacks the lone pair of electron to the deficient carbon and forming a bond with the phenyl isothiocyanate. Since this is the unstable in nature, so it try to break the bond, and consequently it breaks the peptide bonds nearest peptide bond, and that's why the nearest peptide bond becomes weaker. So once you perform the mild acid hydrolysis, so the chances of detachment of the first amino acid is highest as compared to the other amino acid. Hence the first amino acid with amino acid derivatives of the phenyl isothiocyanate is released, and that can be further analyzed by using other technique like the HPLC. So in the same reaction or the same cycle repeated many times, and releasing one by one each amino acid from the N-terminal side. In this way, we can determine the different amino acid sequence from the N-terminal side of a polypeptide. I'll show you the mechanism. You can see here that the phenyl isothiocyanate, because of the high inductive effect of the nitrogen, the carbon becomes electron deficient. So the lone pair of the amine group attacks to the electron deficient side, and then it forms a covalent bond with the phenyl isothiocyanate. But the deficiency is still there, so the extra, but once it attacks to the electron deficient species, this pi bond release their electron and forming the electron to the carbon CO group, because this carbon becomes the electron deficient, and in that way it forming a cyclic compound that is known as thiazolomine derivatives. But it, and in the same time it weakens this bond. So once you perform the mild acid hydrolysis, it is automatically released. So the PTH derived amino acid that only vary in the R group and identified by the automated equipment. So this is the whole mechanism of the Edman degradation method. So protein fragmentation, we can use the different enzymes, and that gives internally cleaves at the different position of the polypeptide chain. Like for example, if you take the trypsin from the bovine pancreas, it cleaves specifically at the C-terminal site of the lysine and arginine residue. Similar, for example, if you have a polypeptide having 10 lysine and arginine residue, it means there will be 11 fragments will be produced. As hence the trypsin is specifically cleaves only at these residues. So you will get idea that if you take a polypeptide, how many fragments and what kind of fragment of what size you can get. Then the another is the submaxillary protease, that is derived from the submaxillary glands, and it specifically cleaves at the C-terminal site of the arginine residue. Then the chymotrypsin is also widely used protease from the bovine pancreas for the fragmentation of the polypeptide, and it especially cleaves at the C-terminal site of the hydrophobic residues such as phenylalanine, tryptophan and tyrosine, because these three are the aromatic and highly hydrophobic in nature. Then the other enzymes such as the Staphylococcus aureus VS protease from the bacterium Staphylococcus aureus, it cleaves at the C-terminal site of acidic amino acids such as aspartic acid and glutamic acid. Then the aspartyl N protease, that cleaves from the aspartic acid and glutamic acid again, but at the N-terminal side instead of the C-terminal side. And then the another acid protease is the pepsin, that is derived from the porcine stomach, that cleaves at the N-terminal site of leucine, phenylalanine, tryptophan and tyrosine. Then the other endoproteinase lysine C, that especially cleaves at the C-terminal site of the lysine, and the cyanogen bromide, it cleaves at the C-terminal site of the methionine. So you need to know four of them: trypsin, chymotrypsin, pepsin and cyanogen bromide fragmentation method. And these all four enzymes, or one is the cyanogen bromide is actually chemical, these are widely used for the protein sequencing methods. I will show you that if you want to sequence a bigger peptide, then the data becomes very hard to analyze. So the best way to cut this peptide into the different fragments by using the trypsin or any other protease. So here the example of trypsin, you can see here that the trypsin is actually an enzyme which cuts the C protein or the polypeptide at the C carboxyl terminal of the arginine and lysine. So you can see here that there are two different sites for the trypsin fragmentation, and it produces different types of fragments once you treat this any polypeptide with the trypsin. For example, here this is a example polypeptide. If you cleave with the trypsin, you can see the number of lysine or number of arginine residue, it breaks at all the places, and accordingly it will produce the fragments. So these fragments now can be used to analyze for the using N-terminal sequencing. So one by one you can determine first you determine the sequence of fragment, followed by the sequence determination of the whole polypeptide chain. I will show you that some reagents that specifically cleaves the polypeptide chain at the certain position. So this is, for example, use of the cyanogen bromide. Cyanogen bromide cleaves the C-terminal side of the methionine. So if a polypeptide having 10 methionine residue, then and treated with the cyanogen bromide, so it will produce 11 fragments. Here you can see here how the cyanogen bromides actually cleave the mechanism of cyanogen bromide derived cyanogen bromide based degradation of a polypeptide. You can see here in a polypeptide that containing methionine residue. So the methionine residue having the lone pair of electron, and the cyanogen bromide, this carbon of the cyanogen bromide is attached with the two species which having the minus I effect, like the nitrogen and bromine. So their electrons are delocalized towards the bromine and nitrogen side, so this carbon is slightly electron deficient. Hence, the lone pair of the sulfur of the methionine can easily donated to the carbon of the cyanogen bromide, and ultimately it forms a species, a cyanogen bromide derived species. Once, because this becomes a species becomes unstable, so it can easily release the bromide ion, and the methionine can forms easily a stable compound of the cyanide derivatives of the methionine. But it gives to the positive charge to the sulfur, it still remains the unstable species, so it tried to cleave from some places where the electron can easily be donated, and you can see here the electron of amine group can be donated in the way, and ultimately it cleaves the polypeptide chain and release the two fragments separately. So this is the mechanism of cyanogen bromide derived fragmentation of amino acid, and cyanogen bromide specifically cleaves at the methionine residue of a polypeptide chain. I will show you a combined method for the determination of protein sequence. For example, you have a polypeptide, okay, and it having one disulfide bond. So first you hydrolyze and the whole polypeptide with the highly acidic condition like the 6-normal HCl, and then your conclusion is that this polypeptide having 38 amino acid residue, trypsin will cleave the 3 times and 2 lysine to give 4 fragments, and cyanogen bromide cleaves at the 2 methionine to give the 3 fragments. So from the amino acid composition analysis, you should have the idea that the polypeptide is of 38 amino acid, in which the amino acid composition like that having 5 aspartic acid, 5 alanine, 2 cysteine, 4 aspartic acid, 2 glutamic acid, 1 phenylalanine, 3 glycine, 2 histidine, 3 isoleucine, 2 lysine, 3 leucine, 2 methionine, 3 proline, and so on. So you can see here that since this amino acid having 1 arginine and 2 lysine, it means it having the 3 potential site which should be cleaved by the trypsin. Then you see there it having 2 methionine, it means it is cleaved into the 3 fragments by using the cyanogen bromide. So from this amino acid composition analysis, you should have an exact idea that if you treat with the trypsin, what will be the possible outcome. If you treat with the cyanogen bromide, what will be the possible outcome. Then from the fluorodinitrobenzene, hydrolyze and separate the amino acid. So you got the 2-dinitrophenyl glutamate was detected. It means the C-terminal amino acid is the glutamic acid residue. So now you cleaves with the trypsin, you have the 4 fragment. One is this, you can see T1, T2, T3 and T4 fragments are there, and T2 placed at the amino terminal side because it begins with the E. It is clear that you can see that the through N-terminal amino acid sequencing, you should have idea that N-terminal amino acid E. So the which one is the first fragment you can easily detect. Then T3 placed at the carboxyl terminal. T3 placed at the carboxyl because it does not end with the arginine. So now the two outcome was derived. Now it is treated with the cyanogen bromide. So you have the 3 fragments C1, C2, C3. C3 overlaps with the T1 and T2 are allowing them to be ordered. So combining all these things, you can actually derive what is the actual sequence of a polypeptide by combination of all different techniques. And I will show you a typical example that despite of the chemical basis of the sequence determination, the sequence can also be determined from the DNA. For example, to find in the beginning of the gene, you can apply the universal genetic code to get the translation product of the gene, that is the amino acid sequence. As technologies advance, it turns out that the DNA sequencing can easily be done over 1000 nucleotide from a DNA fragment, and that have been successfully automated, that high throughput lab sequencing having enormous length of DNA day by day. So you can see that although the chemical analysis is very easy, but it is applied only to a certain sequence length. Because of the data becomes more hazy and more complicated if you increase the, if you want to determine the more number of peptides. So this is the another way around. You can, it is better to determine the gene sequence that can easily be determined, and the gene sequence is translated into protein. Like you can see here, the CAG encodes glutamine, TAT encodes tyrosine, CCT encodes proline, ACG encodes threonine, ATT encodes isoleucine, and TCG encodes tryptophan. So in the same way, if you have the gene sequence, it can be easily translated to the protein sequence by using the simple genetic code method. And this is the now most widely used technique for the sequence determination of a polypeptide chain. I will show you the most advanced and most useful technique for the sequence determination, because all these techniques have certain limitation. Of course, this technique also have the limitation, but this is the most widely used technique for the sequencing of a polypeptide chain. So you can see here in the slide, the first you take the polypeptide chain, whatever you want. So before going to this, the polypeptide is first treated with the trypsin, so it will give you the different types of fragments, and then you can take that fragments and load to the mass spectrometer. So due to the deflect, you can see here that it is, if you generate the high voltage, the molecule is deflected, and it is the collected, the deflection time is actually collected at how long it will take to get from their origin site to the to the detector. So the time is measured, and you know the time is actually, the higher the mass, time is it will take more time to deflect. So there is a correlation between the time of flight with respect to the molecular mass. So first we run the standard of the different polypeptide chains, or you can say the different amino acid, that how long it will take to deflect from the origin site to the detector. And then you can load the another unknown sample, and then you can see that how long it will take. Similar, in the same way, the mass of the unknown compound or unknown polypeptide is determined. Here you can see in the third panel, that is the peptide mass fingerprint. For example, if you have a polypeptide of the 100 amino acid, you treat it with the trypsin, so it will produce many fragments. So the what kind of fragments you are getting, that can easily be detected. The x-axis is showing you the mass, and the y-axis showing you the intensity. So the peptide mass fingerprint is first obtained. Now that each polypeptide is separated using the HPLC, and then each polypeptide separate individually broken, hydrolyzed using the very high acidic condition, and you can see that the probability of breaking of this polypeptide in the different way, and ultimately it produced the fragments of the different size, and the whole data was analyzed by the software that present in this equipment, and ultimately gives you the sequence of each fragment. Once you get the sequence of the fragment, then you can run the blast, and you can find that what are the actual sequence, and then you can what are the actual protein it is. So it is widely used for the identification of the polypeptides from the different proteins. So this is all about the mass spectrometer procedure for the sequencing of the amino acids. So as you know that once the organism evolved, that actually at the molecular level the sequences are changed, and the sequences are changed because to accommodate into to survive in the different conditions. So the evolution is actually based on the genes, and of course gene encodes protein. So the protein sequence is actually the one factor or one essential component that can help you to identify or to understand the process of evolution. So you can see that the effect of evolution not only in the whole organism but also in the molecules and DNA. And for mutation to give effect on the phenotype, and of course it is the subject to the natural selection, and it must affect the structure and function of a protein. So you can learn here that about the evolution by studying the structure or the sequence of the protein, because sequence guides the structure and structure guides the function. So all these things are having a correlation with each other. Ultimately, if you know the sequence, then only you can determine the, or you can understand the evolution process in the better way, while generating the phylogenetic tree using the sequences of the related protein from the different organism combined together and derive the information that how the things are evolved together. So the evolution of the population may occur through a positive or negative selection, or through the neutral fixation of the protein function variant. Then the protein of the different species having similar but not identical sequences, means although the same protein from the two species although having the similar function, but they may have different in the sequences, and these differences are occurred mainly due to the natural selection process, and this fact implies that they have a similar but not identical protein structure. It means at the once the changes occur at the sequence level, it leads to the structure level as well, but although it maintains its function. So Gilbert maintained that the exon represents a structural component of the protein that can be recombined in the different context as a mechanism of generation of the new protein folds. However, the introns are present there, although they don't have function, but during the course of evolution, the changes occur in this intron and they may have some role in the evolution process. So this suggestion could not be supported below the protein domain level, but in the global level, the protein sequence is main factor for the determination or understanding of the evolutionary process. I will show you that how the protein evolved and how long it will take. At the time scale, you can see here the different protein evolves at the different rate depending on its function. It have the different types of origin. You can see here that the histone H4 is conserved throughout the evolution. It is not much changes even during the course of time, because its function is particularly different and it is widely used fit in all the organism. However, you can see as you come to the cytochrome, it evolved at very high speed as compared to histone, and then further the hemoglobin, then further the fibrinopeptides. So here you will get idea that the different proteins evolve at a different rate. I will show you that if you want to analyze the sequence using phylogenetically, so for the phylogenetic analysis, you first perform the multiple sequence alignment. The multiple sequence alignment give you idea that how the amino acids are changed during the course of evolution. And you can see here that the table of alignment of amino acid sequence is very useful tool for evolutionary studies and provide more information than a structure does. The pattern of variation at the amino acid level gives a clues of the selective constraint operating in the sequence or even in the protein structure. So the all conserved region are highlighted in the different color, and wherever the changes occurred in the residue, it is shown without highlighting. So you can see that the different, once you align the different proteins sequences together, it will give the idea that and compare each with respect to one another, and you can find the how the sequences are changes during the course of time and ultimately give rise to the different proteins. So the multiple sequence alignment is the first tool that will give you the basic idea that how the sequences are evolved during the course of time. I'll show you that how the phylogenetic tree further derived from the multiple sequence analysis data. Like it is possible to construct phylogenetic tree derived from the tabulation of the related sequences as earlier I shown you through the multiple sequence alignment. Then phylogeny is derived from the different families of protein from the same range of species are mutually consistent with the branching order. To infer the phylogenetic relationship between the species through genes, it is important to choose the functionally equivalent protein. Means you can compare, or you can, if you want to do the phylogenetic analysis, you have to take the similar types of sequences. You don't take the sequence one protein from the cytochrome and one of the hemoglobin. So it doesn't make any sense for the evolutionary analysis. To have the evolutionary analysis, you must have the sequence having the same kind of function. So you will get idea that how this same functioning protein is evolved in the different species during the different course of the time. So one of the hypotheses have gained much attention is the molecular clock hypothesis, which suggests that the amino acid substitution proceeds at the constant rate within the protein families. So phylogenetic analysis, there are two important things. One is the taxonomy and one is the phylogeny. So taxonomy is actually gives you idea about simply the classification of the organism based on their phenotypic structure or its, so it is mainly used for the organism. But phylogeny, it is the evolution of genetically related group of the organism, or it is the study of relationship between the collection of things, gene, protein, organs, and are derived from the common ancestor. So here you can see the process of evolution of one organism after one organism, or one gene after one gene during the course of time. However, in the taxonomy, it is based only on the, it is the simple classification of organism. So the question is arise that why phylogenetic analysis is essential and what is the role of phylogenetic analysis, why people study phylogenetic? Because to find the evolutionary ties between the organism, how the two organism or more organism are related with each other at the level of evolutionary scale. Then it also helps you to understand the relationship between the ancestral sequences and its descendant, means the ancestral means the old sequence with respect to the newer sequence, and how these two are related and how the changes occur especially at the molecular level. It also provides you idea that the estimated time of divergence between a group of organisms and to share a common ancestor, that how long it will take to convert or to evolve from one to another. So it can be analyzed only through the phylogenetic analysis. What are the simple way to analyze the phylogenetic analysis? Like the starting point, first you set the homologs, align the DNA or protein sequences, and results of the process, the tree describing the evolutionary relationship between studied sequences, that is the phylogenetic tree. You can see that how the tree construction proceeds. So first you consider the set of sequences to analyze, do the multiple sequence alignment properly, check the gaps, check the deletion, checks the insertion carefully, and then the apply phylogenetic tree method. There are many phylogenetic tree methods like the like the bootstrapping, the neighborhood joining method, and the other methods. So all the methods, whatever method you can select, you can apply, and then it will produce a consensus tree, and that tree can further evaluated statistically to obtain the best phylogenetic tree. So this is the simple method for the derivation or for the preparation of the construction of the phylogenetic tree. I will show you a basic example that how the speciation occurs. For example, or you can see that what are the difference between the homolog, paralog, and ortholog. These three terminology are always confusing. For example, you see here that an organism or an ancestor having the A gene, and during the course of evolution, A is duplicated and produce a gene called B. So it having the both A and B. Then during the course of further evolution, the A and B are further deviated into the two different forms like A1, A2, B1, B2. So now the original ancestor is A, which gave rise to through gene duplication gave rise to B, and then A and B give rise to four different sequence species like the A1, A2, B1, B2. So you can see here what is the homologs. Homolog is actually A1, B1, A2, B2. All are homologs because they have common origins. Then A1 with B1 and A2 and B2. A2 with the B2 is considered as a paralog because both they having their common origin, but not all origins are common. So the paralog they evolve at the same time. Then if you talks about the ortholog is actually A1 versus A2 and B1 versus B2 are the ortholog of each other. So these three terminology are always confusing, so you must care of these. What are the homolog, what are the paralog, and what are the ortholog, and this figure clearly tells you to analyze what are these three things. Simple example of the phylogenetic tree showing that different organisms evolve at the different type, like the archaebacteria having the different origin, then the bacterial having the different origin, and the eukaryotic organism having the different say. It is described by the Pace in 2001 that a tree of life based on a small subunit ribosomal sequences. This tree shows that the main tree branches described by Woos and colleagues. This tree referred as the life of tree or the universal tree that clearly discriminates the evolution of the different types of organism like the bacteria, archaebacteria and eubacteria. You can also see a typical example of the phylogenetic tree, and you can see how the phylogenetic tree is actually analyzed. You can see here that it starts with the one from the lower side. It starts with the one ancestor, and it is speciated into the two groups. Then it further branches into the different groups one by one, and you in the right side you can see that the different species it is evolved then in the different time scale, and ultimately the final species is the fungi, that is the eukaryotic organism. So you can see here from the earth, from the very primitive organism to the newer organism are evolved in the different time scale, and ultimately the eukaryotic system was evolved in the form of fungi. So students, let you know, summarize that what you learned from the today's module, that protein sequence comparison is the most powerful tool available today for inferring the structure and function from a sequence. Because of the constraints of protein evolution, a protein fold into the functional structure. So the protein sequence similarity can routinely be used to infer relationship between protein that last shared common ancestor 1 to 2.5 billion years ago. The concurrent development of the molecular cloning techniques like the DNA sequencing methods, rapid sequencing comparison algorithm, and computer workstation have revolutionized the role of biological sequence comparison in molecular biology.